Hello everybody, thank you for watching another video on qualitative and quantitative analysis of pharmaceutical excipients. Today I'm going to talk about sodium laureth sulfate, and this video is beneficial for people who are doing reverse engineering of pharmaceutical products. Before I continue this slideshow, I would like to briefly talk about what is qualitative and what is quantitative analysis. Well, quantitative analysis is simply to determine the amount. So you like to figure out how much of this excipient is present in the formulation. And the amount usually is reported as percent weight per weight. People who are doing science in universities or other field, usually they report the amount as molar or number of moles. But in pharmaceutical field, it always reported as percent weight per weight. So it could be percent, could be ppm, parts per million, parts per billion, parts per thousand. But for reverse engineering, the excipients are present in percent, parts per hundred, and this is how they are reported. The qualitative analysis is about grade determination, the grade of material. So what is grade? Grade could be number. You can define it in a number of ways. One way is to say if they are compendial material or not. That is if they are listed in USP NF or EP or cosmetic. And manufacturer of these excipients, if they are going to define them as let's say USP grade, they have to test them against USP monograph. And by meeting all the requirement of NF, then they will say that their raw material is an NF grade. So this is one way. The second one is the manufacturer grade. Now the excipient could be even non-USP, or maybe they are listed in the IID, FDA IID database or not. But manufacturer could offer the raw material under their own specification, saying we have 4 different type of this excipient, and they have different characteristics. They're probably good for maybe toothpaste, or this one is good for the gel product, and so it would be the manufacturer grade. The third type of grade probably is the USP grade. Let's say USP grade raw material that is manufactured by different manufacturer. So there are maybe 4 or 5 different manufacturers making this excipient, which is, they call it USP grade, but they are not entirely similar. And then we could have a combination of the above. So as a person, as a chemist who's doing reverse engineering, you are trying to figure out pretty much what grade of excipient was used in that formulation, or even you try to pinpoint the manufacturer, like who actually manufactured this excipient. You try to acquire that and you make it use it in your formulation. You will not always succeed in doing this task, but most of the time you will figure out a grade that you could get from your supplier, which is similar to what it was used in the product that you're trying to do reverse engineering. Not too often you will get lucky to get the grade and the manufacturer of the raw material exactly right, unless the raw materials is something very trivial. Let's say glycerin, that it really doesn't matter who manufactures it. Okay, now go back to sodium laureth sulfate, or SLES. This is the anionic surfactant that is mostly used in cosmetic. Means shampoo, soap, toothpaste is a surfactant, and also is used in pharmaceutical dosage form at a low concentration. It is This molecule that we see in the middle has three parts. We have the sulfate component at the left. We have the polyether in the middle, and on the right we have the dodecanol, which is a fatty alcohol attached. This is a heterogeneous molecule, so the polyether chain could vary. So you have a mixture of these molecules. According to FDA IID database, there are different grades of this material. They could be sodium laureth 2 or 3 or 5, and I'm quite sure there are other grades, let's say 8. And also according to the IID limit, they can be used in the shampoo or in the semi-solid or solid dosage form. As I'm quite sure that the SLES does not have a USB monograph, so it is not a USB or NF grade, but it is listed in FDA IID database, and it has been used in approved drug products. For qualitative and quantitative analysis, I'm going to talk about HPLC with the ELSC detector and GC. The chromatogram that we see here is the chromatogram of a dosage form that I was reverse engineering, and the SLES is shown as a multiple peaks between 4 up to 10 minutes. And the earlier peak that we see in this chromatogram are the peaks related to the other excipient. So with using SLES, we can do quantitation. We can determine the amount of the SLES present in your product that you're trying to reverse engineering. And also because you have multiple peaks, you have a profile that using the profile you can determine the grade. You can get information about what grade of raw material was used in the product that you are trying to reverse engineer. The chromatogram was generated by using C18 column, and the mobile phase is water or buffer with organic solvent. Is a gradient program, and because the SLES is elute in a very wide range of retention time, it is very likely that in other formulation that you want to reverse engineering, you will have the interfering peaks. So this method may not be suitable for all products, but it happened to be suitable for this particular product that I was working on. The next technique is the GC method, and this is in this method you only will be able to get the amount of the SLES. There would be no grade determination. In this technique you will do the acid hydrolysis of the entire finished product, and you will break off the dodecanol. And by using the GC, you will determine the amount of dodecanol, therefore you will calculate the amount of SLES in the formulation. This is useful technique when you have major interference with let's say HPLC with SLES detector, that you cannot do the amount determination. So using the GC, you will then be able to quantitate the SLES by determining the amount of dodecanol. There is a small peak around 9 minutes. It could be other fatty acid present in the formulation, so you need to have a placebo to determine that. And at least you have to keep an eye that maybe SLES has other fatty acid in it, so you will look for the undecanol or decanol or other fatty acid as a minor peaks. There are many, again, the references for determination of SLES are too many. I only listed one of them that talks about the acid hydrolysis and the GC. Thank you very much for watching this video and have a great time.